All right. So as Batul mentioned, I'm an assistant professor at Michigan State University. I'm a computational biologist, and we develop computational approaches to study infectious diseases. So that's my day job. What I also like to do is, wait a minute, there is a little bit of an echo that I'm not able to avoid. Can you hear me better now? Okay, great. Okay, so what I also love to do, what I'm passionate about, is teaching education and outreach, and especially to promote and provide a safe space for underrepresented minorities in R and data science. So today we are going to get started with our workshop on Intro to R and Tidyverse. I see from your initial poll that most of you are actually familiar with R and maybe even a little bit of Python and Jupyter Notebook. But today we are going to focus on one suite of packages, which is exceptionally helpful when you're doing any sort of data wrangling or data visualization. And while the workshop itself is not focused on genomics or transcriptomics, we'll use a processed transcriptomic dataset just to demonstrate and learn everything about Tidyverse. So I hope you can still see my screen. And if you have trouble, please ask us questions on Slack. There is a channel starting with Tidyverse. Okay, here is how the GitHub repository would look. And if you're trying to run these codes locally, what I would recommend is you download the zip file and then you have a local copy of it. And once you open the local copy of it, this is what it's going to look like. It'll say Workshop Tidyverse wherever it is that you've downloaded, whether it's a Mac or a Windows system. And then you can just open this R project. It'll automatically open an R studio with this as your home directory. So just open the R project. Yeah, so that's all that you really need to do in terms of setup. And I hope you've installed your necessary packages. If not, you can directly go to the install packages part and just run these two commands. Okay, and while you set up, I'm just going to get started and go over my slide deck. Okay. And there is another link that I'll share here. This link is also on Slack. I've shared this on chat. It's a fast cups link. See if you can access it. I would encourage you to please log on. So if any of you have trouble, I would encourage you to actively use this. So for example, if everything is going well, green is your color. If you kind of are struggling or something, then I would say go for yellow. But if you're completely stuck and you really need me to stop, then you're going to hit on red. So on an average, if it's mostly green with a little bit of yellow, I'm going to keep proceeding just in the interest of time, since we also started a bit late. But if I see more reds than greens, then I'm going to stop, take your questions, and then proceed. But also after each section, I'm going to stop to take questions. I want this to be as interactive as possible, so stop me. But let's also keep an eye on the clock. And if I don't keep an eye on the clock, Batul, please help me here. All right.
I'm keeping track of all the questions in the chat here and inside Slack, so don't worry about it.
Excellent.
Thank you, Janani.
Okay. So the raise your hand, I don't know about the raise your hand thing now because it's not Zoom anymore, but I would still say ask your question on chat or Slack or whatever it is. Batul is going to monitor it. Stickies, I will monitor it myself because I have it open on another screen. Okay. I hope you have our studio Tidyverse installed. Otherwise, please install it in the background. So today, what we are going to focus on is the suite of packages, Tidyverse. It's beautiful. It's very elegantly written by Hadley Wickham & Co. So it has multiple packages. So there are packages to read, import, and export your data. There are ones for tidying up your data. I'll tell you in a little bit what I mean by tidy data. And then there are a few packages for data wrangling and data transformation. Then there are a few packages for data visualization, which is really the fun part of R. Modeling, which we will not touch upon today. And finally, all of the code that is on GitHub that you can also open up is on R Markdown. And it's all about reproducible research and writing your code with documentation, with code, with the output and everything in one document. And that is R Markdown. If you're familiar with Python, the closest equivalent would be Jupyter Notebooks. So these are roughly the 5 parts that I've labeled here, and I'm going to go over one at a time. The most time-consuming parts would be dplyr and ggplot. So after dplyr, I'll give you a short break, and then we can get back to the data visualization. Okay, so if you have RStudio and you're using the default colors, you might have noticed that mine looks black because most of my things are in black. I prefer that. So this is what your RStudio is going to look like. I'm guessing most of you are familiar with it, so I'm not going to dwell too much here. If you're installing Tidyverse and then loading it, if everything looks good, this is what it should look like. Again, the white version of it. Okay, so the first step is how do you import your data? So the way I'm going to do this presentation is I'm going to start with a few slides, and then I'll go back to RStudio, and then you can follow along with me. Okay, so for instance, for the following up part, like I said, you can go to the so for instance, if you've opened the R project, this is where it will lead you. You go to the transcriptomics because that's what we are going to focus on today. And then if you open this R Markdown file, it's going to look exactly like it looks here for me. Okay, so this is on the back end, but we'll get to it in just about a second.
Janani, someone asking
Yes.
this sorry to interrupt you. She said, started saying she missed what we're supposed to download from GitHub. I don't see a zipped file. Would you mind to re-illustrate that step if you don't mind?
Okay, I'm going right back there. So this is what the full GitHub repository looks like. So if you look at the README, it will have all the information. Look at it later on if you haven't already looked at it. But what I was recommending that you do so that you have a local copy is you click on code, you click on download zip, and then you unzip it. Once you unzip it, this is what it's going to look like. You'll have a local folder of it after you unzip the local copy. And then what I was recommending that you do is open the R project file. When you open the R project file, you don't have to open any other file here. Don't try to go into transcriptomics here. We'll do that within RStudio. So once you download, unzip, open the R project, go to RStudio, and here you go to transcriptomics and the R Markdown file. That's the order. If you want to open the corresponding HTML, you can just open the HTML, and this is what it's going to look like. Or there is also a web version, and I've shared this link with you. I've also shared this with you on the Slack, platform Slack. So this will have the corresponding version with the codes, with the output, wherever there is output. So whatever you're comfortable with. Okay? All right. So let's get back to the presentation quickly.
Thank you, Janani.
Oh, no problem. If anybody is still having trouble, please also, I only see green cups right now, so I think I'm good and I'm going to proceed. Okay. All right. So like I said, the very first step is, well, install the library that you've loaded, which is Tidyverse. It's going to help pretty much with all of it today. Here are a few basic commands that we'll be using again and again, or you will be using even going forward, which is to import different formats of data. So the standard, if you're used to base R, you might notice that there are equivalents of this called read.csv or read.dlm. So the read underscore, all of these underscore functions usually are from Tidyverse. But these days, most other packages, that deal with tidy data also have this format. Okay. So read underscore CSV basically it has the default parameters that are set to read a CSV file. So all you need to do is really point to where the file is or the full path. And then you just say, if you have column names, you just say col names equals true, and that's it. If you have an unspecified or a weird delimiter, because you don't know where you're getting the data from, right? Then you just use the read underscore delim, which is much more general, and you can use the delimiter that you want to use. So basically, read underscore CSV would essentially be, instead of this double slash, if you have a comma, it's going to be similar to the read underscore CSV. There's also a beautiful package by Jenny Bryan for Read Excel. This is for absolute R novices who've never used R but want to migrate from Excel to R. So you can actually use the read underscore Excel package. You can specify what's the file, but you know there are a lot of complications with your Excel file, right? You might have merged lists, you might have multiple sheets. So there might be only one range of cells that really has clean data. So what you're going to do is you'll just specify which sheet, what is the range, and do you have column names or not? And it's going to import it cleanly into R.
Sorry, Janani. I got also a few questions. I think people, when they downloaded the zip file, they don't know how to open it. Like, say, they don't know where the code is. It's a bit lost inside the whole repository. I see like 5 people asking the same.
Okay, let's go back right to that. So is it fair to assume you don't have a folder like this, or have you downloaded, or do you have a zip file?
It seems like they do have a zip file, but they don't know where the code is.
If you unzip it, you're going to see something like this. Is that right? Okay. So once you have a local folder, in your case, it might be in downloads. You go to downloads. Sorry, my downloads is a mess and it does not have this. But you'll have a zip file, unzip it. You'll see workshop Tidyverse. Then what you do is you double click or say open, or open with RStudio, whichever works for you, depends on your defaults. If you say open, it's going to open this R project file in RStudio. And how do you know that it worked? Well, in the top right, where it shows you whether it's part of a project or not, here it will say workshop Tidyverse. If it says nothing, then it means you've not opened the R project file and you've just opened RStudio by itself. If it opens in the console and if it does not open in RStudio, well, there is a good chance you don't have RStudio installed. But if you do have RStudio installed and the R project does not open in RStudio, but you have RStudio, I would recommend that you use open with, and here you click on rstudio.app. Maybe if you check here, your default might have been R. That's why it's opening it in the console. Or you can just say other, and here you can force open it with RStudio. This is to absolutely make sure that you're opening the R project file with only RStudio and nothing else. And now you should again see this here. If you go to files, you'll see so this is going to be your home directory. So your working directory will automatically be set to workshop Tidyverse. The beauty of opening the R project is, doesn't matter what is upstream, because this is going to be variable from my computer to yours. But everybody is starting on the same, you're starting at the same place, exactly the same place, the workshop Tidyverse. That's why I'm encouraging you to open the R project file.
Someone just asked,
can zoom in.
Yeah, thank you, Janani.
All good? Okay, maybe it's too zoomed in, sorry. Okay, I hope you can read now. And if anybody actually has accessibility issues, sorry, I should have mentioned this upfront. If my dark colors are really upsetting you a lot, I can switch it back to the default. So just let me know. Otherwise, this is just here for my preference. The zip file will be in download. You unzip it. you open the folder, you open our project with our studio. Zip file will be in download unless you have a different default place to download it. Or you can just go to Spotlight if you're using a Mac, Command and Space, and just look for where is workshop tidyverse.zip, and it will show you where it is. And you just go right there if you can't find it. And if everything else fails, what you can do is basically use the link that I shared with you on chat. That should have the full presentation, and you can follow along and we'll troubleshoot with you. Let me see, where is the majority? I still see a few reds. I still see a few yellows. Okay, yellows, speak up. Let me know what the problem is, or let Batul know. Otherwise, I still see a majority of greens. And if you don't mind, I'm going to move forward. But I really hope you've at least gotten to the stage of having our studio open. Oh, wait, if you have our studio open, here's another thing you can do. You can just say open. You can just say open file, and then you can go to the folder and also open the R project. These are different ways to make sure you're opening the right file in the right platform and you're in the correct.
Yeah, just one last thing, because some people, it seems like they download it, but they don't know what to find as a file. I'm just posting now a link to a Google Drive where I downloaded all the repositories. So thus you have all the repositories there. If you're not sure how to download it from GitHub or where to find it, just that's where it is. Yeah.
Excellent.
Okay. Thank you, Janani. You can go on. Yeah.
Okay, great. Oh, it seems like I have so many of them now. Okay, so this is a brief intro to today's data set. Well, since you're an ISCB, I'm assuming that you have a little bit of interest in biology, even if you don't have background in biology. So we are going to deal with an RNA-seq data set from these authors. The cool thing about this data set, it has multiple samples. It has a few different platforms. So there are multiple technologies here. There are different tissue types, and there are different life cycle stages. So we have a few different variables to play around with, which makes it a perfect. So the code base was developed by Arjun and me. Arjun is also an assistant professor here at Michigan State. So this is how your samples and data would look like. And I'll show you in just a second how it's going to look like once you load the data set into R. Now I'm going to go back to RStudio. So you can ignore this first part. This first part is supposed to render here nicely, where it basically gives you an intro about me. It shows you how to install RStudio, R, how to install these packages. I don't think this is a problem anymore, but in older versions of R, people had trouble with installing Tidyverse. In that case, I had them install these packages individually. So all of you good with installing? This is a specific FastCup question. Excellent. Most of you are green here. Sorry. So all of you have installed Tidyverse. Anybody having trouble with installation of Tidyverse or loading Tidyverse or here? When you load the library, this is how you load. You go to library and Tidyverse. Let's go here. Install packages, loading packages. So if you have the R Markdown file open, how would you do that? You start here. You go into transcriptomics. Sorry, is this big enough or is it too big? Okay. Are you able to read, all of you? Okay. So you go into transcriptomics and you click on this R Markdown. Here is where you are. If you want the outline and we want to go to specific portions, you can go here. I'm going to loading packages. If you want to run the entire, what's this called, chunk. If you want to load the entire chunk, then you'll just hit on play. But I would recommend you go line by line. You can use Control Enter if you're on Windows, Command Enter if you're on Mac, and it's going to run it line by line. It noticed that all of these are comments, so it directly went to the next viable function here. How can I see the code instead of just the console? If you click on R Markdown here, so if I close this, you'll only see the console. But if I click on R Markdown here, from my Files tab, in Transcriptomics folder, then this would open up. Genevieve. So you'll have to click on the R Markdown. Okay. So you're here, you've loaded Tidyverse, you've loaded the here package. This is again a really cool package with pretty much one function. Here. And this is again by Jenny Bryan. It basically makes sure that we are all in the relative same space. So when I run a function, when I run something, we are all starting with this workshop Tidyverse and everything downstream. So my codes are also going to work for you. I don't have to worry about where you have downloaded it, which platform you're working on. It is going to work regardless. So that's why here is extremely helpful. And as I use it, you will see how it looks like. So this is our Markdown. If you're familiar with any amount of Markdown, you will see that this is where I write my general text. Yes, somebody had unmuted.
Yeah, sorry, Janani, if you just can zoom in a bit, it's a bit small.
Okay.
That would be great. Yeah, brilliant. Thank you. Yeah, it's better.
It's better? Okay.
Yeah, I think it's better.
All right. Okay. So let me give you also a little bit about R Markdown here. So as you can see, there are different parts in R Markdown. So you have text. For example, here I've said data import. And then I just have a bulleted list, and this is how it's going to render. So now that you have loaded the here package, if you say here, if you run this, what you should see is where is this workshop Tidyverse in the grand scheme of things on your computer. For me, this is where it is. Okay. And then the next thing I'm going to run is I'm going to start with basically this data set. Again, you don't have to download it because we have a local copy of it, but I'm going to have you download just so you learn how to do it. So if you run this URL, it basically is where the data set is. And then here, I'm just going to say this is a tab separated file. This is not a comma separated file. So I'm just going to use read_tsv from the readr package. And this is the assignment operator, which says, okay, I'm reading the file, but I need to put it in some variable, some object that stores the contents of this file that I'm reading in. So once I run this, I'll see there is a new object here that's created on the right. Can you see my arrow? And it says this is this huge object. And I'll show you a few different ways on how would you look at this object. If you don't know what read TSV does, or for that matter, if you don't know what any function does, all you'll do is just a question mark and read_TSV, and the help page will show. And these days it shows up actually rather nicely. It used to be all black and white earlier on. So it will show you what the different options are. These are the other related functions, the CSV, CSV2. CSV is for comma, CSV2 is for semicolon. Okay, and a single question mark would suffice if you have the package already loaded. If you don't have the package loaded, it might say, I don't know what you're talking about. In which case, if you at least have it installed, you can use a double question mark and it's still going to pull it up. Okay, you just do a double question mark and it will show you what are the matches for that, and then you can click on it. This is in case you've not loaded the package. But these are quick ways to check the help documentation from within your console or your code. Okay. So these you cannot run. These are more of mockup functions because I just say path to my data and I don't have folders called path and to. But this is how you would read a file if you had it locally with read_CSV or read_DLM. I showed you these on the slides. And in this case, you can also read the file that is because it's here locally available. Well, it's supposed to be. Okay, this one, the RNA-seq counts. But you don't have to do it because we just downloaded it from the URL and you're good to go. So you do have the local file here. All right. Does everybody have this local object? Now we are going to start looking at what the data really looks like and what am I talking about. So I mentioned that there are different technologies. There is an adult and a fetal, different life stages. So we want to see how does all of this look in the data set. Please slow down. I'm sorry about that. Okay. I will try to speak slowly. So you have the data set. The first thing you're going to use is a native R function, which is called just the structure. So when I run that, it just shows me what the data set really looks like. It says this is a data frame, which is fair because we imported it with Tidyverse and it loads it in a data frame format. It shows you what the different columns are and what is the what is the data type. It's either character or in this case numerical. But this is a little hard to read. Don't you agree? There are so many things going on here. And then it shows you what are the data types again. Let's see if there's a slightly better way to look at it. Let's just see what happens if you type the variable name by itself. This is already better, right? That's because we loaded it with a cleaned up data set. And there is a typo here. Okay. So there are these different columns, but there are so many more columns beyond the scope of my little window here. So it says these are my other variables. And it shows you what are the data types. Again, it says these are character ones. These are double. These are numerical ones. So you see there is a gene ID. There's the chromosome, the start and end location, which strand of the chromosome it's on, what is the approximate length of these different genes. And then these are all the different samples all the way to the end. So there are 25 columns and there are 57,000 observations. So this is again a different way to take a quick look at it. Now I'm saying head. Head basically shows me just the first few rows. The default is 6 for R. If you're using this on command line, your default for the head command would be 10. But it doesn't matter. You can change the default here. If you want to see 10, you can see 10. You would just say n equals 10 and it's going to show you 10 instead. Okay. So what this shows me is I don't want to look at the whole thing. I first want to know what my data set looks like. That's always the best place to start. You need to know what it looks like, exactly all the quirks, all the good things and the bad things about your data set. Is there missing data? Is it unclean? What kind of cleanup step would it need or cleanup steps would it need? You need to first figure that out. So the best way is to just open up your data set and explore it even before you actually type out any commands to analyze or manipulate it. Okay. So these are a few other ways. You can check all of these out. Glimpse is yet another way, which just gives you a quick snapshot. It shows you what all the columns are and what are the first few entries in each of them. If you just want the column names, it'll show you what those are. If you want to know what are the dimensions, you can also look at that. How many rows, how many columns. If you want to subset the data frame, saying I want only the first 5, I want the first 5 observations. The first element is a row on the columns, and then I want 7 to 10. So basically, I'm starting here with the samples. So see, 7. So it shows me these. It starts with the colon, and it goes on to liver. So you can subset the data set this way. And if you want to write back a subsetted data set, you will use the complementary version of the read underscore TSV that you just used. You would just say write underscore TSV, and it's going to write the data frame. Okay. So you will say, this is the variable that I want to write. I don't want to write the whole thing though. I just want to write a subset of it. You tell it what the name is, and that's it. It's going to write a local copy of it. And if you write it, it's going to save a subset wherever it is that you are. And then there are a few other packages like Knitter. Knitter actually might also be part of Tidyverse, but I'm not entirely sure. If not, when you try to load it, it's going to complain. So you will know whether something is installed or not right away. For me, it didn't complain, so I have it installed. Now this looks really, really ugly. And you would ask, why am I even showing you this? Well, the reason I'm showing you this is because when you render it in your HTML, now all of these are very hard to see, right? All the stuff that I showed you on the screen, but here it shows me in a nice tabulated form. And this is just the head, so it shows me only the top 6. But I'm not entirely happy with this because I want to see my full data. So then I would use something called a page table, which is in R Markdown, which is in R Markdown. Again, R Markdown is part of Tidyverse, so you don't have to load it again. And because it says paged table, it just takes you through multiple pages. It has all the columns here. The entire data is here for you to explore. And all of this can be seen just on the HTML. So this is a way of how you would render the data using R Markdown and HTML. So it's just a matter of this one line command. So nothing complicated that you have to do. Isn't it fancy? Okay, everyone still with me? I'm going to check the green cups. Ah, good. All right. So now I'm going to go back to part 2 that got done pretty quickly. So the next part is, how do you tidy up your data? And what do I mean by tidy data? So the second part is using Tidier, which is again another package within Tidyverse. So we call a data set a tidy data set if each variable has its own column and each observation has its own row. It is as simple as that. If it is clean cut that way, then we say that is a tidy data set. For example, here, each variable, country, year, population, each of these variables have their own columns, and then you have multiple observations. And what is in each cell, as you would call it in Excel, each cell, that's the value. So that's what is the observation for this variable country, that's your value. So this is already a tidy data set that we are looking at, and this is what we want to bring data sets into. So for example, this is a wide data set where you have all of these spread out versus I can convert that into key value pairs, saying for each variable, what is the value? So I take this, for example, we saw there were 25 columns, right? And especially just if you take the samples alone, they were going from column 7 to 25. There were a whole bunch of them. So there were almost 18, 19 samples alone, right? So but they all have exactly the same kind of data. So in those cases, it actually makes sense to convert it into a long format where I say, what is the sample name and what is the value? And that makes it much easier for me and much easier for your downstream analysis when you bring it to that format instead of reading it in this wide format that you can't even see on your screen. So there are very easy ways to flip back and forth, and I'll tell you what is the use of that too. So the most common functions that we will use in Tidyverse are pivot longer. This used to be called gather if you've used Tidyverse in the past, maybe 1 or 2 years ago. What this does is takes all the columns and it gathers it into the rows. So it says pivot longer. It goes from the wide format to the long format. And then if you want to put it right back, or if you have a long format and for some reason you want a clean looking data set, then you might use pivot wider. It used to be called spread. And then there are 2 others where you can do some basic data wrangling with respect to your column operations. So if you have one column and you want to split it across multiple columns, your data, then you would use something called separate. And then if you want to, again, just do the opposite, you would use unite. In case you're wondering, when would you use something like separate? When would I want to separate one column into another? Let's see if I have an example. Okay, I don't. So for instance, if I have year, month, date, right? So I have the 4-letter year hyphen month hyphen date in one column. Now that's great. Sometimes I might want the date if I'm plotting some continuous or categorical data. But sometimes I might want to know what the sales are, or for a particular year or for a particular month across years. Now, there is no way for me to get to that if I keep the data column intact. So in that case, what I might want to do is to separate it. I would say take this one column, you find those hyphens, separate it by that, give me 3 different columns instead. And now I can do the operations by year or by month. So that is when it becomes extremely powerful in using these simple one-line functions, separate and unite. I'll again demonstrate all of these to you, so it doesn't have to remain theoretical. Okay? This is basically how gather would look like. Gather is basically pivot longer, and spread is pivot wider. These are old animations from Garrick. He has beautiful animations in Tidy Explain repository. You should totally check them out. So again, in just cartoon format, this is what your data would look like. So if you have these different columns, you will see that those are being converted into key value pairs here in pivot longer versus wider. And same way, this is the year example that I just told you about. You're splitting it into year, month, and date. And then you're bringing it back, saying you join them together with a hyphen. Okay, everyone's still good? I still see a lot of green cups, and I'm going to proceed. I still see 1 or 2 red cups. If you have a question, please post them either on Slack or on chat for Batul to look at. And I can help you when if you have a question. Okay, now we're going back to RStudio. All right, so this is what I just showed you in part 2. If you want to jump right to part 2, what you will do is you'll go here and you'll say, I want to go to gather spread. Okay, I'm closing the outline. Okay. So by the way, here is where I'm naming the chunk. I'm saying that this code chunk is in R. Actually, you can even say Python here, because you can use Python or any of your other programming languages also within the chunks. I'm just using R. And then I'm saying, yes, please show this piece of code when I render it in my HTML, which is why here it actually shows it here too. And then I also say evaluate it as well, please. And then it evaluates it and also puts the output there in my HTML. Okay, that's what the echo and eval mean, in case you were wondering, because this is going to come up again and again. So I'm not really spending a lot of time teaching you R Markdown, but since everything is written in R Markdown, here and there I'll point out a few things. But if you have questions, again, in part 5, we can focus on this part. All right. So let's get started with tidying up your data. So you have this data frame here. You saw what it looked like. It had more than 50,000 rows. It had 25 columns. Now you want to see what you can do with it. So the very first thing that I want to do is, well, you've already looked at the data. So if you select anything and you just say Command Enter or Control Enter, it's going to execute only that part of the code. Okay. So in this case, I have this whole bunch written here, but since I selected only GSC ID, it runs only that part. See, it only runs that part. What is this operator? It's actually a beautiful operator called the pipe operator. Now native R also has an equivalent that you can use. But well, Tidyverse was using it for quite a few years before then, so I'm going to continue to use this for today's workshop. So what the pipe operator basically does is it connects these different functions. It strings together them beautifully. What do I mean by that? You can just read the pipe operator as then. So for example, you can read this code as start with this data set, then you select these columns, I'll show you, I'll tell you what I'm selecting it by. Then you either gather, or see, gather is the older version of it. Now it says pivot longer. Then I pivot longer. So that is how you read the pipe operator. It seamlessly takes you from one step to another. Okay, so in this case, let's see what the first step does. What select does is it basically subsets the columns. Filter works on your rows or observations, and select works on your variables or columns. So here what I'm saying is, okay, so this is what my data set looks like. But I want to subset my data, but in a very specific way. I want to keep the gene ID because, well, that's my identifier. I don't want to lose that. But I don't want all this other information about the genes. That's kind of the metadata at this point. I don't want that for my current purposes. Instead, I want to just keep the sample read counts alone. So how do I subset that? Do I want to spell out these other 18 columns? Well, maybe not. There is an easier way to do it. What I'm looking for is a simple pattern here. I said it is either adult or fetus, right? So it's either A underscore or it is F underscore, but it's always only one or the other. So if I just do a simple string pattern matching, I say if it matches either A or F, that's why it's within a square bracket. This is called a regular expression, or regx. If you're familiar with other programming languages, maybe you've used something very, very similar there too. But all that I'm telling is look for either capital A or capital F followed by an underscore. And please pull up only those columns. So what happens when I run it? See, you just have those 20 columns. You've dropped all the other ones that you're not interested in right now. You have the gene ID, and then you have all the sample columns. Now I'm saying, now it's still a wide data set. I still have a lot of columns here that are essentially, it seems like these are all different variables, but actually they're not different variables. They are different kinds of the same variable, which is a sample. They're all actually just samples. So I can convert it into a cleaner, longer format, which just says, this is a gene ID, this is a sample, and this is the value that corresponds to the sample. These are the counts for it. So what am I going to do? I'm just going to say, this is fine. This I want to leave intact. So what I'm saying is, you take the columns that match this pattern and you convert it into a longer format. Remember that animation where it goes from wide to long? And then I say you convert it into a sample counts pair. Let's see what happens when you run this part. See? For example, you'll see this is all the same gene ID. You'll see 19 of those or 18 of those. I don't remember the total column number. Okay, you'll see 19 of those. So for every pair of sample and counts. Let's see. Everyone with me so far?
Yeah, no questions so far. Thank you, Janani.
Okay. And we are also going really slowly. That could explain why we have no questions. Okay, good. So now if you just want to go back to the original data, so up until this point is what we have already done, right? So this is what I just did up here. But then I'm adding pivot wider and saying take the sample and the counts and put it back. And then I get back the original data set. So pivot longer and pivot wider are essentially complementary functions. Now let's look at unite and separate. So you may have noticed that there is a little too much going on in these sample names. There are actually 3 different pieces of information here. So what are the different pieces of information? One part is telling you whether it's adjuvant or biochain or this origin, right? That's a different technology. And then the second letter is telling you whether it's adult or fetus. And then the last part after the underscore is telling you whether it's colon or heart or kidney. So you have all of these things going on in the sample name. So it's a little complicated, don't you think? Sorry for going up and down. Right now I'm back here. I'm also going to undo in case I added a few lines, and I'm going to go back to separate and unite. Okay, I'm on line 241 of the R markdown in case you lost me somewhere. Okay. So what I want to do here is, again, I want to do the same thing as before, which is select only these columns. And yes, I want to pivot. So far, we are still doing the same thing as before. So far, you're still with me. But then here we are doing something really cool. I'm saying I'm going to split the sample column into source and stage and the tissue. And I know in this case that my separator is the underscore. Let's see what happens. Well, something should happen. Does it? Sorry, it's hidden behind my laptop. That's why I'm jumping around. See, now you have your source stage in one column and you have the tissue split into a separate column. This is really cool. So now if I want to look at heart or kidney or tissue samples across these different stages, I can do it, right? It was just this one line that I had to add to split it. Now I'm saying, wait, there's actually two pieces of information here. I want to separate it again. And here I'm saying separate after the first character. So there are different ways you can, if you look at the help page, you can actually look at this, right? So there are different ways in which you can separate. So here we used a delimiter, but here I'm saying separate after the first character. And let's see what we get. Okay, we have more than 1 million rows, so it explains why it's a little slow here. There you go. So you have your source, stage, and tissue all split up nicely. And if you wanted to go right back, you would just do everything until 257 line. It's the same as above. But then I'm adding unite. Okay, I'm not combining all of it. I'm just combining the stage and the tissue, which is a different combination than it was before. See? Whether it's adult and heart. So I'm pulling out the source, where the data comes from. I'm just combining the biologically meaningful information together here, which is which life cycle stage and which tissue. Okay, and with that, we are going to part 3. And we are still behind time, I think. Or not. I think you're okay.
I think it should be okay. And I did extend the time just in case we go over. So don't worry about that, Janani.
Okay, great. So any questions so far? Because we are moving into part 3. Now might be a good time to ask any TidyR related questions. All right. So next we are going to look at an extremely powerful package called dplyr. It was built based on Plyr. And it's very, very helpful for basic data wrangling. So you've already looked at select, which we use to subset different columns. You can use so many functions within select to exactly identify which columns you want to subset and how. Same way, filter is also very powerful. You can use a whole bunch of Boolean operators and you can use certain conditions and say, this is exactly how I want to subset my observations or rows. Then there are 2 more functions, mutate and transmute. Mutate is very helpful in creating new variables based on existing variables. So if, for example, the simplest case you can think of is you have 2 different columns and you want to multiply or divide. You have some annual information and then you want to convert it to a monthly, right? So you can create a new variable based on existing variables. Transmute works like mutate, except it drops the original columns. Arrange is the tidyverse equivalent of sort. So you will use it to sort based on a different combination of columns. You can say maybe one of it is in ascending, other is in descending. You can specify all of this and arrange your data frame in a particular way. Summarize and group by are very helpful when you're working with different subsets of your data and getting summary statistics for not just your whole data, but for different subsets of this data. And we'll see what that looks like. So filter is basically you're subsetting different rows. Select is when you're subsetting different columns. So when you use summarize, for example, if I just say mean or average, right, for my different samples, for instance, for each column, I would just get the summary statistics. But instead, if I say group by first, meaning it groups like data into subgroups, I can specify based on which column. For instance, if you take our own data set, maybe I want to group the adult samples and the fetal samples together. Or maybe I want to group it by the tissue type. So colon samples, but across source and adult or fetus, right? So I can group in different ways or even different combination of these columns. And then I might want to create summary statistics, not just for the whole, because that might not be informative at all, but I want to get the summary statistics for each of the groups. So usually when used together, group by and summarize can be very, very helpful operations. There are other functions too in dplyr that I'm not going to be talking about, but feel free to explore. Again, there are cartoon versions of all of this in Tidy explained by Garrick. So there is the left join and right join and inner join and outer join. These are basically different ways in which you can combine data frames. Whether you're keeping all the columns and all the rows in the first one or the second one, and how are you combining them? So based on that, you would use these different functions. So for example, this is what left join would look like. It keeps all the rows from the first data set. And right join, it would keep 1, 2, and 4. It keeps everything from the right or the second data set. Inner join is it would keep the common ones. And well, as you can expect, the full join would keep all of them, which might result in some missing data, yes. And then there are set operations. So for example, this is the only one that matches fully. So this is intersect. And union is it keeps all of these different combinations. And then if you have similar data frames, meaning the columns are the same, I can use bind underscore rows. And actually, if you are binding at all, I would strongly recommend that you use bind underscore rows because it is more cognizant and it will throw you errors if they are not exactly looking the same. If there is anything not right, your column names are not the same, your formats are not the same, it's going to complain and you need for it to complain. So you don't make the mistake of combining data frames that should not be combined. So use bind underscore rows or bind underscore columns and specify exactly how you want to combine them, by which row you want to combine them. Okay, with that, we are going back to do some data wrangling of our own. So,
Janani, yes. Sorry, while you are showing how to do the data wrangling, if you just go back a step, someone is a bit confused about the data. Like we are using now tab delimited files, but he was thinking, how did you go from BAM file to the TSF? I think he just not sure if you can elaborate about data formats and how you use it inside transcriptomics analysis.
Okay. If I understand right, you're asking me how to get from the tab limited data format into R into all the other operations that I'm doing.
Yeah, so that's the question. I've just posted it from the Google Doc into the chat.
No, this was not a BAM file. I've done the transcriptomic analysis. I've converted it to a counts table. I'm not showing you here how to do the transcriptomic analysis, but just saying that if you've done a transcriptomic analysis and you have a tab limited file, which is the counts table in this case. If you have that as an output of your transcriptomic or RNA-seq analysis, that is what you would input here. So for example, let's see if we can look at the raw file. This is just... Can you read now? Sorry, I forgot that it was not readable.
Yes, good.
Okay, this is it. Let's see. Okay. So this is what my input data looks like. It's tab separated. Well, it's supposed to...
It looks good.
Yeah. So this is what my input data looks like. And this is after I've done the transcriptomic analysis. It has all of this information about each of these gene IDs. And then it has the counts for the different samples. This is what the raw counts table is going to look like. And then I'm bringing it into R using my read_TSV, because I already know that the delimiter is tab. And when I bring it in with read_TSV in the data import part here, or you can, if you have a local copy like I just opened up here, you can also use this. You can say file. Here is the file in transcriptomics, and this. You can say it's TSV because it's tab separated. And yes, keep the column names. And when you open it up, this is what it's going to look like. Okay, so that's the input file. Then I converted it into a longer format. And then after that, I hope you're with me. Does it answer your question?
That's brilliant. Thank you, Janani.
Okay. So let's start with filter. Again, as a reminder, this is how your full data set looks like before I remove a few columns. So here what I want to do is I want to just try filter a little bit. I want to say take the whole data frame. And then I only want to look at genes that are smaller than 50. So if they're less than 50 base pairs, those are the only ones that are showing up. And there are only 124 of them of the 57,000 that we started with. So this is how you would write a nested function. But since we like pipes, we are going to say take the data set, then filter. Of course, it does exactly the same thing. Now I want to do something else. We have all these different chromosomes, right? Now I want to say I can also do a regx or a substring search. I can say take the chromosome column instead. And if you have used any programming language, you might have used different variations of grep. So here I'm using a logical grepl. I'm saying if you find chromosome Y or genes on chromosome Y, return those for me. And there are about 540 of those. I could say, you know what? I want both. I want the chromosomes on Y, genes on chromosome Y that are really small. So I would run both of those at once. And well, there is only one that exactly matches that condition. And what I used here is the Boolean AND. You've already seen one instance of select where we used capital AF within square bracket followed by underscore, right? Now I'm just showing you, I might keep saying regx. What I mean by regx is regular expression. So if you just Google regx followed by R or R and regx, you'll see a whole bunch of how to use regular expressions in your R codes and which functions you would use along with that. But if you're familiar with programming, well, you might have used regx even in other programming languages for pattern matching. That's what you would use it for. Okay, here what I'm going to say is I want to match this particular pattern. I'm saying yes, again, keep the gene ID. I always want the gene ID in this case because that's my identifier. And I'm saying match only those that have F underscore. But there is this little dot here. Now, okay, so this is not an interactive platform. I was going to ask you, do you know what the dot is? Since you may not be able to answer, I'll tell you what the dot is. So the dot is to say any character. And here I'm saying any single character. So like you saw earlier on, it could have been one of the 3 different sources, right? So I'm saying it doesn't matter to me which source is it. As long as there is this one character, it can be anything, bring up all the matches and it brings all of them, whether it's AF or BF or whatever it is, it brings all of those columns down. And there are only actually 5 of them, including the gene ID. So this is another way to match. You can also select by saying, so earlier on we said everything that matches either A or F, right? But what I could have also said is instead, remove all of these. In this case, there were only 5 to remove, so it is fine. I could have said it. But sometimes it can get clumsy because you don't know how many you have to remove or what their exact names are, which is why it's better to use a pattern matching and say, but you're going to get exactly the same results though. You'll get the same 20 columns. Whether you say just keep these or just remove these, you're going to get the same result. And you can do that too in select with just a simple minus. If you want to do just the flip side of it, saying, hey, I don't want to see the counts, I only want to see the metadata. Instead of just saying matches, you would just say minus match. Now you only have the metadata, just these 6 columns. Now we know how to string together functions easily, don't we? So what we're going to do here is we're going to select a few columns and then select a few rows. We're going to put that all in one workflow here. So you start with the data set, then you select the chromosome and the length. Because why am I doing that? I'm doing it preemptively because I have some operations here based on the chromosome and length. That's why I'm keeping these 2 columns. And then I'm keeping everything else. Then I'm saying I want only those that are less than 100 and on chromosome Y. I wizened up. I used 50 earlier and I got just 1. So now I'm saying I'm going up to 100 and it has brought up 22. Okay. I still see mostly green.
Yeah, everything good.
Okay.
Yeah, nothing the Google Doc or the Slack. Yeah.
Okay. So then we are going to mutate. What mutate does, like I said, is if you want to add additional operations, additional columns. So here, again, I'm just selecting everything but the counts and head just to see what it looks like. Well, this is what it looks like. More typos. Okay, so here what I'm doing is I've selected this. So far you've looked at it. Now what I'm saying is, see, it says gene ID. So mutate, the way the structure is, it says this is the column I want to operate on. And this is the new column that I'm creating. In this case, I'm just going to replace it. That's why I've not given it a new name. And then I'm saying what is the operation that I'm performing on it. In this case, it's not a mathematical operation because it's a string, right? It's a character column. Instead, I'm going to clean it up. How am I going to clean it up? In this case, let's see what this does. Okay, I don't want to reveal here what this does. Let's just run this up until here and see what happens. Oh, it's running it on everything. Okay. So basically what G sub does, it substitutes. So I'm looking for a particular pattern. and I'm substituting it with a different pattern. So if I just press tab here, it's going to say what is the pattern and what is the replacement. Right? So what I'm looking for is a particular pattern here, and I'm saying replace it with nothing. So what's the pattern I'm looking for? I'm looking for dot. Earlier I told you that we use dot to specify any character. But in this case, I'm actually looking for a dot. So how do you tell that to R? Saying, hey, I don't mean any character, I mean the actual dot. So I'm escaping the character. This double backslash is basically escaping it. I'm saying now I'm looking just for the dot itself. And it could be followed by any digit, any number. In this case, I'm only seeing 4 and 2 and 1. But well, it could be 10 as well, right? So I don't want to say just one character after it, but any number of characters, which is where the star comes into the picture. And this dollar is saying towards the end of the string. So the caret sign, which is above 6, can you see it here? In my console, this caret is to specify it's the start of a string, and the dollar sign is to specify it's at the end of the string. So what am I really looking for here? What I'm saying in this simple cleanup step is I want to take the dot followed by any number, not just any digit, but any number, because it can have multiple digits. And I'm going to replace it with nothing. So what it should essentially do, well, it should have done something. I don't know what is going on. Oh, because I've saved it to something. No wonder. See? Now it has basically removed whatever is after the dot. So that's all that I've really done here. Then I want to attack the next column. So here I'm seeing there are so many of these because there are multiple start and end fragments. So I'm going to take, for this purpose, I'm only going to take the first one. How do I do that? I'm saying anything that is after the semicolon, any character, any number of times. That's what the combination of dot and star are extremely helpful to use together. Dot says any character, star says any number of characters, right? So I'm saying anything that follows pretty much after the semicolon until the end of the string, I want to remove. So what does this look like? Now it doesn't have anything after the semicolon. So all of this is gone. It just says chromosome 1. Well, now you know how to do it for start. I'm doing exactly the same thing. I'm just keeping the first corresponding entry for each of them. So I'm doing the exact same operation also for start. But here I'm just telling it, after you do all of this, now it's a character because it has a semicolon and everything, but eventually convert it to numeric. Because I want to do some operations on the start and the end coordinates. I want them as numbers. I want to be able to subtract. If they're characters, I can't. So I'm just being wary of it upfront. I'm doing the same for end. I'm doing the same thing for strand. Now let's see how this cleaned up file or data frame looks like. So I've saved it to this variable called gene loc, which is basically the location information of the genes on the genome. It says which chromosome, what's the start and the end coordinates, which strand, what is the length. So I'm just giving it a different variable. I'm just keeping the first entries because we want to do something downstream with it. There you go. Looks much cleaner and simpler, doesn't it? And you'll notice that it's not character anymore. And I deliberately changed it here. Okay, still with me? Now I just do head. Or I basically clicked on this, which is the same thing, just to look at how my data frame has changed. Now that I have something that's not just string and a whole bunch of string, in fact, that I don't want, what I now have is something I can perform operations on. So now I can convert this to KB. Right? This is in kilobases. I can create these new variables with mutate. See, it says 11.9, 12.2, 1.76. But it keeps the original columns. If I use transmute instead, it drops the original columns. This might be helpful, right? I don't want to use that information. So I'm saying keep the gene ID. I mean, don't do anything with it, just keep it. Okay, now I've not lost any information. I've added these columns with KB. Okay. Now I'm going back to gene lock, not all this other stuff that we have done. So just as a reminder, this is what my gene lock looks like, the variable. I'm saying distinct chromosome. So I just want to see how many distinct or unique values my chromosome variable can take. It is 25. Guess why? There are 23 chromosomes, right? Sorry. Because it has your 22 somatic chromosomes, then it has your X or Y, and it has the mitochondrial. That's why it has 25. Well, my strand is pretty straightforward. It's just plus or minus. I can also pick combinations of them. So it would basically have 25 into 2. So I see 50 rows. I can arrange them. So it starts with Y. I'm saying descending because this is character. If it's numerical, I can just say minus. For example, here, I'm saying sorted by chromosome and then sorted by length. If I say minus length, it's going to start with the longest one. Minus here doesn't make so much sense because this is a character column. The minus makes more sense for the numerical ones. Otherwise, you'll just say descending or ascending. Okay. So we are done with arrange. Next, for the really cool part of group by and summarize. So what would happen if I just group by these different, now you saw there are 25 chromosomes, right? What do I mean by grouping by chromosomes? It says now there are 25 groups because there are 25 distinct values that my chromosome column can take, right? So it's not doing anything to your data frame yet. It still has that. It's just saying, though, it just has in mind that there are these groups. Now what I'm saying is get me the number of genes. This is a simple function that you can just use to count. And then show me what the first gene is. How do I know which is the first gene? I get the minimum of the start. Let's see what that looks like. Isn't that really cool? I didn't have to do anything complicated. Right? All I had to say is group by your different chromosomes. And then you summarize, tell me how many genes are there in each chromosome. And where does the first gene start? Now, if I didn't have this, what would this look like? It just tells me where the very first gene on the chromosome is and what the total number of genes are. This is also helpful. But you can see why using group by gives you this additional piece of information and the additional resolution in your data analysis, right? Okay. Now let's do some more fun things. Now what I want to do is arrange it by length. So start with the smallest one. Then I want to group by chromosome strand. We've run all of these individually. So there are no surprises here. I'm starting with the data frame. I'm doing all of this. All of this you already know. You're starting with the tiniest ones. Then I'm adding a whole bunch of summary statistics here, or actually just basic information. I'm saying what's the smallest gene, right? And then the minimum of the length. And then some basic quartile information. What's the median of the length, which is the longest gene. And the cool thing is I can do this for each chromosome and for each strand with just this small piece of code. And if you want to write this, by the way, all the stuff that you've done here, you've not saved any of that yet. In fact, you've not even written it to a variable. You only have these two variables. But let's say I want the gene location information saved separately in a file. I'm just going to say write underscore TSV. Again, I can use the pipe. I don't have to put it in here. I'm saying here, so it takes the relative path. And since we are in the project and since we are here, automatically it's going to be in workshop Tidyverse. It's just going to go one in transcriptomics and it's going to save it here. And it's called the gene locations. You still with me? Is now a good time for a break, Batul? Otherwise, I can do this more data wrangling and we can take a short break afterwards.
Yeah, I think we can take after that. We are keeping with time. We have now 90 minutes left, which is good. Yeah, so
Okay.
if we can.
That's great. All right. So let's do some more data wrangling now. Now the assumption is you're familiar with the basics of how to filter columns, how to filter rows, how to arrange, how to identify what are the distinct entries in each of the columns. You know how to group. You know how to summarize by groups, how to add these summary statistics. You know basics of regular expression. Already you've learned quite a bit. Now let's see what more we can do with what you've learned so far. Okay? And just as a reminder, this is what the data set looks like. I don't expect you to remember everything about the data set. So here and there, I'm posting some redundant information in some sense. Let's also look at it just in case. So this is what it looks like. Okay. So what are we doing here? First, we are going back. It seems like we are going back to counts. So we are removing all the metadata. We are going back to the A and F and all the sample files. Okay. So we are doing a few things here. It seems like, let's see. I don't know if that works. So you have, okay. So you have your stomach 1, stomach 2, and stomach 3. Wonder what those are. So for the time being, I'm saying, I only want to keep one stomach for the OA combination, which is, I think, origin adult. Okay. I don't want multiple. Just for this purpose, I'm not saying don't throw out all the data that you don't like. That's not what we're doing here. I'm just trying to teach Tidyverse. So we are saying I'm just going to keep stomach 1. We'll worry about stomach 2 and stomach 3 later. So for now, I'm just removing it. So all I'm saying is equals null. Okay. And then I'm doing what we did before, which is I'm not going into which version of it. I'm just keeping the gene ID itself. And let's see what the simple cleanup looks like and how many columns we have. Now you have an additional variable here in your environment, gene counts. You have 18 columns because you've dropped the stomach 2 and stomach 3, and the stomach 1 has been renamed. Also, your gene ID has also been cleaned up. That's all that you've done so far. Now, what am I doing? I have my raw counts. So what I want to do is to convert it into log CPM. CPM is counts per million. So I'm taking the counts, basically the counts that you have in each of these columns by the total count. I want to make sure I don't encounter zeros because I see a lot of zeros here. It is a sparse matrix, right? I'm converting it to log CPM. I'm removing it so I can say mutate all. Okay, that's the only reason I'm removing this. So I don't have to specify regx. I don't have to do that. I can just say do it on every single column. And I get my log CPM. What does it look like? Basically, these are my log CPM values for all the samples. And as you can note, there is no gene ID column. Well, I can't make sense of it, can I? Anyway, these are some simple statistics that I can do because it's a fully numerical matrix at this point. The minimum, the median, the max, the quartile range. Here we are using our bind calls because we are saying, I actually want to put back the gene ID before I lose track. Maybe if I rearrange something, then I'm messing up the whole order, right? I don't want to lose that. So as soon as I convert it to log CPM, I want to take the gene ID column alone and combine it with the log CPM and put it in a new variable called log CPM. But I'm also doing other things here. So this we have already done, right? Converting the sample into source and stage and tissue. And then we are saying remove because then it removes the original one, which is called sample. Okay, let's do this one by one. I don't want to complicate things. Okay, so far you're with me, right? By the way, it says gather, but this is pivot longer. I'm sorry, I missed this one here. But it's the same as pivot longer. So you have it in the long format, you have the samples, you've seen all that is different as it used to say counts earlier on, now it says log CPM. That's the only difference really. And then what I'm saying is I want to separate the sample column into source, stage, and tissue. And I'm actually saying remove false, which means keep it. If I wanted to remove the original one, I would say true. Then what am I doing? It says underscore column because I just did it by character position, right? So I'm saying if you see at the start, remember the caret is the start and the dollar is the end. So I'm saying if it starts with an underscore, Replace it with nothing. I don't want that. I just wanted to directly say colon. And then I want to add one more thing. Here it just says A. That's not very informative, is it? Especially if I'm going to analyze my data and make plots. So I'm going to spell it out. I'm saying if it is A, call it adult. If not, it's an if else. So the first condition is if it's A, call it adult. If not, else. The other only option here is F. So it's going to say fetus. Now let's see what happens. There you go. We still have the sample. We have the source. We have not done anything to it. The underscore is gone. And now it says adult. Okay. View is going to basically pull it up for you here. But you don't have to do it. You can just look at it here in the console, whichever you're comfortable with. Okay. And before we go to the visualization part, just as a teaser, let's just look at one plot before you take your break. And then you can come back ready to attack all plots. So now we have the log CPM. You know what the log CPM looks like, right? What I'm doing is, I'll tell you what I'm doing later actually. ggplot is basically the package, the function, the everything that allows you to make beautiful visualizations in Tidyverse. GG because it's grammar of graphics. And I'll tell you what those are right after your break. But what I'm doing here is I'm basically plotting my sample, my log CPM by tissue on stage. I'm making a box plot. Let's see what the box plot looks like. Let me give it some room too. So if you generate a plot, it's going to open up automatically in the plots tab. Okay, maybe I didn't give it enough room. I'm going to try it again. Okay. So if I don't give it enough room, it's just going to not plot. There you go. I specified the color is by tissue. So you have this legend here. I'm saying the line type. So it's either full for adult or dashed line for fetus, and it automatically does it. See, this is why I kept my sample column, because I didn't want to plot it by tissue. I just wanted you to use these for the different colors and the different patterns. So I can specify how I'm plotting this, but I just want to plot the samples by themselves. And it shows me what the log CPM distribution looks like for each of these samples. With that, we can take a break.
You are muted, Janani.
Yeah, okay. So one of the things that I can see is that you've replaced X with the actual variable name. So I would still say you're using mutate all, which is to say do it across all the columns, but you're replacing X with a particular column name. So you're kind of giving it two different contradictory pieces of information. You either say mutate and use it on only one column, or you say mutate all and do it across all columns. So that's the first problem that I see. And the second problem is we have already replaced OA stomach 1 with OA stomach. So they might not be OA stomach 1 anymore. So this might anyway not work for you. So I would just say do the mutate all in this case because you want to do the same log CPM calculation on every single column. So do that. And I'm guessing that this will work. And to the other person's question, Shruti. So gather, in case gather is not working for you, Let me share my screen again. Can you see my screen?
Yes, we can.
Okay. So for example, it used to be gather. I replaced it with pivot longer because that's the newer version. And it used to say key and value. I've changed it to names and values too. And then this should work. Okay.
Like
And
I'm
I'm
about
sorry, I
to
forgot to
show
replace it.
my face somewhere.
Marvelous, you're unmuted.
Marvelous. Is it working for you? Okay, I think.
Okay.
Okay.
All right. So does anybody else have any questions? Sorry, I didn't look at the Google Doc yet.
I did not worry. There's questions there. I think we're safe to continue.
Okay.
Thank you,
All right.
Janani.
So let's get started now. Let's go back to the presentation. So now we are starting part 4, which is really the most exciting part of R for me. This is how it all started for me. So, well, ggplot is a fantastic package to make all kinds of visualizations within Tidyverse. Like I said, GG stands for Geometry of Graphics. And for any ggplot that you're making, it requires at least 3 elements. The first element is, of course, data. What is your data frame, right? So you first specify that. Then the aesthetics, or it's shortened to AES. How your data is going to be represented visually. Is it a single dimensional, two-dimensional plot, three-dimensional plot? What is the X and the Y? Do you have additional attributes? For example, in the teaser that I just showed you in the box plot, I said the color is by tissue or the line type is by something else. Or if I have points, I can say what is the size, what is the shape depend on. And then finally, and most importantly, I have to specify what is the kind of plot that I'm making. What's the geometry or the geom? In our previous case, I used geom_box plot because I wanted to make a box plot. But you can make a scatter plot or a bar plot or anything else really. So you've got to specify based on your data, whether it's categorical, continuous, numerical, or something else, whether it's time series, right? Based on your kind of data and what you're trying to learn from it, you will decide what is the best geometry to represent it and what are the other customizations that you're going to add. Okay, I'll show you a few different examples now, but you can do so many things, and I'll also point you to some couple of fantastic resources to go after if you're especially new to ggplot or to making plots with R or Python, for example. And finally, hopefully we can make a plot like this, which not just makes a scatter plot for you, it facets it by groups it by continent, and then you also have this animate. Now your transcriptomic data might not be as much fun, but you'll see that in the folder that you downloaded or the repository, there is another folder called Gapminder. I would very strongly encourage all of you to try all the scripts also in Gapminder as you did for transcriptomics. Okay, so it's a completely different data set. So the plots and the cleanup steps might be slightly different. So you'll just get more experience for whatever you already learned today. So for example, let me use the Gapminder data set here to show you what are the 3 elements that I mentioned. So I'm just loading the Gapminder. It's a data set that's available in this package. You don't have to run this now. So we'll go back to the transcriptomic data set in just a second, but I'm just demonstrating a point here. So for ggplot, there are three things I said that are needed. The first thing is the data set Gapminder. Then I'm saying I need to plot what are the X and the Y. I'm saying I want the GDP per capita and the life expectancy. I want to add two attributes to the plot, which is size by population and color by country. Then I'm saying I'm specifying what is a geometry. This is a scatter plot. I want to by point. Alpha is basically saying there is a transparency that I'm adding, right? So let's see what this looks like. So this is what it looks like, right? And here I've added facet wrap by continent. If I remove this line, it would put all the continents together. Now it just helps me see some trends a little better because I've resolved it. I've just wrapped it by continent. This one line basically makes 5 plots for me with shared axes, which makes it much easier to see than just one plot with everything on top of each other. I've also added log scale because, well, it's GDP per capita. It makes sense to look at it on the log scale because it spans several tens of thousands. Okay. Now for the animate part, all I had to add to all the existing code from the previous slide is just to say again, I'm just adding some labels here, but this is really the only part that I'm adding, which is the transition time is year, and it's a linear transition. That's all that I'm specifying. And there is already a year column there. So it picks the corresponding rows, and then it makes this beautiful plot. Okay, with that, we'll go back to RStudio to make our own plots. Yes, like Batul said, we are in part 4. How do you get to part 4? You click on the outline, you go to part 4, and here we are. Okay. So quick recap, ggplot, you're going to hear that a lot. You're going to hear the aesthetics, the geometry, and let's see how to plot our gene location data to start with. So here I'm starting with the gene location data. It's always a good idea to remind yourself of what the data looks like because we have a few different variables here. So, okay, so this is the gene location. I have the gene IDs, the start and the end and the length. I'm just going to make a very simple bar plot. Here by the counts. Okay, I'm just making a geom bar. Now you can see how it's all on top of each other, right? It's not particularly the cleanest plot we can make. Also, let's see if it gets any better. Okay. Also, you may notice that it says chromosome 1. Then it jumps to chromosome 10, 11, 12. Because they all start with CHR1. But that's not the order that we want it in. So what we are instead going to do, okay, so what we're instead going to do is we're going to specify what is the order that we want this in. We specify what are the factors. And we are saying these are the different levels. So forget everything else there. If I just paste this, let's see what this looks like. What I'm doing, I'm just taking the character CHR. Then I'm saying go from 1 to 22 because there are 22 somatic chromosomes. Then X, Y, and M. And then string these together. What this will give me is a string of these 25 chromosome names in the order that I want. That's the most important part. So what I'm telling this factor function is saying, now you just have your chromosome. Well, it's all a little bit of a mess. So what I'm instead going to do is to take the factors and keep the order of these factors. That's all that I'm specifying. I'm not changing anything else about the data. Now I'm making the exact same plot. You see, now it's in the correct order. It ends in M and starts with 1. Still, it's not the most beautiful of plots. You still see a lot of overlapping text. What can we do? There are two things I want to fix. I don't want this gray background, which comes by default in ggplot. And second, I want to flip it and see if that makes it any better. So I'm using two simple functions. One, I'm just saying use the minimal theme, no fuss. And then I'm going to flip the coordinates. And because I saved the plot to this variable here, remember we saved these objects, the data frames. You can also do that with plot variables. So each time I don't have to keep typing this out again and again and again. If I like a combination that works, I can keep the combination and then I can just say plus, plus, plus. So if you remember from earlier on, this pipe function that we used was to string together multiple tidyverse functions. What the plus does is something very similar, but it works within the space of ggplot. So if I want to add different layers or different pieces of customization within ggplot, I would use a plus instead of a pipe. The plus tells me, wait a minute, there's one more thing that's coming on the next line. The same way the pipe tells me, wait, there's one more function that I want to add. Okay, so that's the difference. So when you see a plus, you know there is more ggplot stuff that's coming. Okay, this is already great. I can read it, which is more than I could ask for in the previous plot. And I have a basic simple plot. Now I can add some simple embellishments. I'm saying there's the number of genes in each chromosome. I'm just adding X and Y labels. That's it. We have made our first simple plot. Now I'm going to make a histogram instead. What with? With the length. Let's see what that looks like. Again, I'm selecting only this part. I'm not adding the last two. So I want you to see what it looks like before I clean it up. So this is what it looks like. Right? Then we realize very quickly that probably it's a good idea to add a log scale. Okay, that makes a lot more sense. Again, I want to remove the gray background. There you go. You have a simple histogram. What are the three elements? Reminder, you have the data frame. You're specifying what it is that you want to plot, the aesthetic. Then you're just saying, I want the histogram. That's the geometry. I'm adding two more things to simplify the plot. So next, what are we going to do? I showed you this facetting, right? For example, if you go back to the slides, I showed what the facet does is basically it creates multiple subplots. We want to see if maybe instead of doing this bulk across all the chromosomes, what if I facet it by chromosomes? Right? Everything else is the same as before, but the only thing that I'm adding is the facet. I'm saving it to a new variable because now I've already added several other steps here. That's it, just that one line. And I have the histogram by chromosome. Now the colors look nice, but they're not informative because anyway, they're sitting in their own box. I'm not going to really get confused which chromosome this is, right? Because this is just the chromosome 1 data. So I'm just going to remove the legend. I'm just saying legend position, none, and I'm adding these labs. Again, I'm not changing anything about the plot, so I'm just using the same plot variable here instead of copy pasting this entire 5-6 lines. Okay, it's just a shortcut. Makes life easy for me. I do this. One, it should remove the legend. There you go. It gives you so much more space. And then you have the axis. I've still not given it enough room, so you see that this is not readable yet. So you can change the angle of that. Okay. Next, let's make some scatter plots. And for this, you will use something called geom_point. So you've seen box plot, bar plot, and histogram. This is the fourth kind, which is a scatter plot that you're making. What are you making with? You're still with the gene location data. What I want to plot are two things here. One is the length. We already have that variable. Let's say you already have the length variable. And then I have my start and my end. I'm saying plot end minus start. It's numerical, so it can compute it dynamically. I don't have to compute it, save it in a new variable and call that variable. I can just say end minus start and it's going to plot that. Okay? Then I'm saying, let's just do that first. Let's see what it looks like. Well, it's a plot. There are some patterns. You can see that you have no points here. But it's not very helpful beyond that. You don't even know how many points there are. So we're going to add a few customizations here, few things to make it slightly better. First, I'm adding alpha. Alpha is to increase transparency. You can call this within a lot of visualization functions. Okay, so I'm just saying, well, make it transparent, reduce the size, and I don't want black, make it gray. Now let's see what that looks like. And I'm saving it to this plot variable. This is start minus end versus length. I'm just saving the plot. Well, the gray is getting in the way again, isn't it? I'm going to remove the background again. So we just look at the points. And this time, I realize that everything is clustering there because I'm not using the correct scale. Once again, I'm using a log scale both for the X and the Y. And suddenly a pattern emerges. You can see the data, and it's already so much more helpful than the first plot. Great. I'm glad to see in the chat that at least one of you has the plot. I hope more of you do. Let me see what the fast cups is looking like. You still have the link, right? Okay. Thank you, Batu. So if you're not comfortable with something or if you're getting stuck, let me know as you're making these plots. Okay. So now I'm going to add a few more things here just to see if I can make this a little more interpretable. First, so these are, again, I'm just adding geometries to the existing plot. Now we have a plot. So all of this is going to be carried forward. Okay. I'm just adding more layers here. So what am I doing? I'm adding a slope 1, intercept 0 line. That should basically go through 0. Okay. We'll get to what this means, but you'll already see there is a clean divide. There is nothing here to the right. Then I'm also adding some couple of other intercepts. Okay. Now, what do you think this means? Why do you think there is nothing here? Okay, sorry, I forget that you cannot answer. Or maybe you can answer via chat. Why do you think there is nothing here in the white part and why all of your data is above the red line? No? Okay. So here what we are plotting is basically the gene length against the end minus start. Right? So here we are basically saying the upper limit is what is defined really by a gene length. But you can have, based on the different spliceoforms, based on the different combinations that you see in your data set, your different transcripts, you could see that they might be shorter than that. But it absolutely cannot be longer than the final gene length. Right? So that is what you're really seeing here. So you're not seeing your end minus start ever go above the gene length. And you're seeing this just by plotting your data and looking at it. And yes, it makes sense. You know you're looking at transcript data. These are not just by genes. Yes, Deepak. Now we are going back. We've done this before, right? What you're doing is you're grouping by chromosome. And for each chromosome, you're getting the mean length and the number of genes. Let's plot that. So these are all tidyverse functions, right? ggplot is also part of that. You can just combine them. Let's see what this looks like. I'm just saying, take all of this, whatever analysis that you've just set up here, a simple summary table, and you're feeding that into ggplot and saying now plot the number of genes versus the mean length. Okay, it's not really telling you much. By the way, on line 569, you might have to install the package. I already have ggRipple installed, so I'm just going to load the library. If not, take a second to install this package, and I'll show you what it does. Well, GG usually, most packages that start with GG basically means they use the same grammar as ggplot. Whether it's GG genes or something else, I can show you those additional examples too. But here, ggRipple is basically N. N basically counts the number of entries. So in each group, it counts how many are there, how many observations are there. That's what your N does. So for each group, if it found that there are 5363 observations, it will give you the 5363. That's what the N means. It doesn't take any argument. It just adds up. Okay. So now what we are going to do is, so far, all of this is still what you did. I'm just going to remind you, it's the same plot. That doesn't make a lot of sense that I'm making again. Then I'm adding a little bit of I'm saying, okay, what's the trend looking like here? Just add a smooth trend and an alpha. Some labels to know what I'm looking at. And then there are a lot of points here close by. So I'm just going to use the ggRipple, where when there are really close points, it just staggers and adds these labels so you can read them. Now you know what these points are that you're looking at. So there are only 25 points because, well, there are 25 groups and that's what you're trying to plot. But you couldn't make much sense of the previous plot, so we're just adding some labels. So you're seeing chromosome 1 by a large margin, has the most genes. And probably also has few of the longest genes. The mitochondrial ones, it has very, very few genes. Again, not a big surprise. Same goes for chromosome Y. And they're also relatively, on an average, they are smaller genes. And I always like to use theme minimal so I can look at the data more cleanly. There you go. You have a plot with labels. You're just looking at, again, some simple statistics, but you're looking at it visually so you can make sense of it. So some people can just look at tables and tables and tables all day long and make sense of it. But for others, seeing them visually helps make a lot more sense and interpret the data much better. Also to figure out what other analysis you have to or need not do based on how the data looks like. So visualizations are extremely powerful that way. All right. So I showed you a teaser of a box plot, but now let's make the box plot properly. So you can ask me questions too if something doesn't make sense. And we're going to make a close relative of these box plots, which are the violin plots. For this, because we are looking at the distribution of the counts, we are going to go back to the log CPM. Again, since we are going to switch data frames, I just want to post a quick reminder. This is what your data frame looks like. You have the samples, just so you can call the samples in a concise way. But you also have, for each sample, what the metadata is, whether it's adult or fetal, which tissue it comes from. And finally, for each gene, for each sample, you have the log CPM value. And this is a lot of data. close to 1 million rows. So what's the best way to visualize this, right? When you have so much data, when you have these different groups, and when there is a distribution, clearly, one good way to make a plot is using geom box plot. How would you do it? Well, just take it, specify what you want to plot. You want to look at your samples. You want to look at the log CPM. Let's say I take all of this out, right? And let's just make this. What does this look like? So you know how these customizations are coming into play and why they are helpful. So this is your stripped down version. How does it look? Well, it looks like a box plot. That's what we wanted. And it does plot sample by log CPM. Not what I would call a very helpful plot though. Sure, we can flip it so the labels are more readable. No, it needs more space. See, it doesn't even plot. It doesn't even take me seriously. Okay. Come on. Okay. But now I want to go back and add this. Well, you know very well what theme minimal does. So here what I want to say is this is great, but I'm not able to see if there are any patterns by tissue type or by adult and fetus. So if I also want to see that metadata projected on this plot, I'm basically going to add these two attributes here and remake the exact same plot. There you go. So you see these are the hard samples in the steel. Then you have your adult and your fetus. Your lung is the one that clearly has samples that are going up in terms of log CPM. You're seeing a lot of difference. Still, let's see if we can make it a little more interpretable than it already is. I mean, this is helpful. It's at least more readable, if not helpful. Let's see what else we can do. So in a box plot, for those of you who have not made box plots, this line right here in the middle is the median. You have your 25th and your 75th quartile. You have the other plots, sorry, points. And here I think we've also plotted the outliers. Now I'm going to make a violin plot. Okay, let's see what the violin plot is looking like. Excellent question, Batul. Hope you all have some plots to also play around with. So this is what the violin plot looks like. I'm specifying also what the limits are for the scale. And the nice thing with the violin plot is you can actually visually see the distribution, where it's tapering, and all the little blips that we are seeing with the data. So there are more points here. You'll see all of that nicely in a violin plot. But essentially, both violin and box plots are showing you the distribution. Okay. We can also specify, for example, make the outlier not so bright, not so big. So just add a few more customizations here. Let's see what this looks like. Okay. So I've added a little bit of transparency, made the points smaller, and I've saved it to this variable, sample box plot. Okay. We'll get back to something like this in just a moment, but let's take a little break and make a few scatter plots here. And what are we doing here? We are just going to compare. For example, I said there are different technologies that are essentially doing the same biological kind of samples, right? They're either both adult or both fetus and from the same tissue. So we want to see if there is any variation when you go from one source to another. So here we are looking at fetal colon, F colon, from A and B. I think one is Agilent and B is, I forget what it is, but well, it's the other source. So again, here we know we can seamlessly string together the tidyverse functions. So I'm saying on the go, I'm just taking the data frame. We saw the ampersand, right? We saw and, that was the Boolean operation to string together two conditions and saying, filter, get me the observations for chromosome Y that are smaller than 50. We use the and. But here I'm saying if they're either AF or BF, I'm using the pipe. This is the other pipe. This you'll find on your keyboard below delete. So you're saying either take this or that, which means it gets you all of the samples that are either this AF colon or BF colon. I'm only taking these three columns, gene ID, sample, and log CPM, not all of the other ones. But then I'm splitting it again. So let's see what this looks like. I'm selecting it and then I'm spreading this. Okay, so now I have my gene ID, my AF colon and my BF colon. Okay, now I'm plotting my AF colon versus my BF colon. That's the reason instead of the long format, I've converted it to a wide width spread. By the way, this might be pivot wider now. So in case it throws an error, that might be the reason. And here it says key value. So what you might have to do is pivot wider. This would be names from. Instead of value, it will say values from. Sorry. And this should work. Yeah, it's the same thing. Okay, so in case your gather and spread are giving you trouble, then this should fix it. Okay. And then this is what we are giving ggplot to say plot AF colon versus BF colon and make a scatter plot for me. And we already saw the geom smooth. I'm saying just use a simple linear model and look at this. I've already added alpha, reduced the size. First, this makes me happy. Yes, there's a lot of variation, but at least it seems to be more or less centered around the diagonal, which is what I expect, seeing how they're both fetal colon. If it was trending completely in a different way, it would have been upsetting. So you know you can't really take any source. Your data, your interpretation highly depends on where it's coming from. But yes, there is considerable variation around what seems like a quintessential fetal colon value for each of these. Again, you can save these plots too to the variable. How would you actually save the plots? I'll show that in part 5. But for now, you can just create these variables and then just go on. Now let's do some more fun stuff, similar to how we did the additional data wrangling. Now you know all the basics of ggplot. What does it expect? What are the different kinds of plots that you can make? Well, there are several more, and I'll show you that in a second too. Okay, so now what we are going to do is we are going to get the high variance genes. So how do I do that? I have the log CPM. I'm going back to this one. I just want to see how many total number of genes are there. So I'm just getting the total number of distinct. Why am I doing that? Because, well, I've expanded it, right? So for the same gene, I could have multiple entries for each of the sample in the column. So this does not tell me how many gene IDs they are. So I'm just going to get distinct. And then what are the number of rows? Well, this is what we started with. So yeah, no surprise. It's 57,000, around 57,000. Now I want to calculate the high variance genes. How do I do that? I'm saying group by gene ID because that's what my metric is going to be based on. I'm getting the interquartile range, the IQR. It's a built-in function of the log CPM. And then I'm taking the top entries there. Let's see what this alone looks like. Or even, well, even before that. How many rows with it? 2891. Let's do this. Okay, so now I'm getting the IQR for all of the genes. You're with me so far? I've grouped by gene ID. That's why there are only 57,000. Earlier, we had some 980,000 something, right? Across for each of the samples. Now it's combining all of these samples and getting me the interquartile range for each gene. Then I'm saying, I'm looking at the top 5%, which are the most variable of sorts, right? And let's see what that gives me. Okay, it gives me less than 3,000, and I started with close to 60,000. And what pull does is it basically says, for all the conditions that are being satisfied above, just pull the corresponding gene IDs alone. Because I want to know, want to have somewhere note what are the genes that are the highly variable ones. So pull basically gives me what are the corresponding gene IDs. I could have even said select and just the gene ID, but this is a way to get the character string of all the gene IDs, okay? What is the length? Meaning how many are there? Oh, sorry, I didn't save this. So if it gives you an error, nothing to panic. We all get errors all the time. That's life. That's how programming goes. You just go back, retrace your step and see if you actually ran it correctly. In this case, I was selecting it and running, so I was not saving it to the variable. But well, now the variable exists, so I should be good. Yeah, this is what we saw. It has around 2,800. Let's plot this. Now, what I want to do here is not just look at the high variance genes, but also look at the high variance genes from chromosome Y. So we have this gene location, right? It's the original data frame. We know what this does. It filters only the chromosome Y genes. It probably had a few hundred. And then I'm adding one more condition here. And again, this is a beautiful operator that will really help you in subsetting data. So what I'm saying is, I'm telling it what the condition is. I'm saying I only want the highly variant genes. But how do I know which ones they are? Well, that's why I did all of these things here. And that's why I've pulled the gene IDs. So I have that stored. So what I'm saying is subset, show me only the genes that are in this high variance gene list and show me only those that correspond to chromosome Y. Well, there are only, 14 of those. And I'm pulling the gene ID again. And I'm saving this now as not just high var genes, but also chromosome Y high variance genes. And now that I have this stored, Well, I can just do this in one step going forward, can't I? I'm just saying, now I'm pulling this gene, these genes, but now not necessarily, sorry, I'm pulling these genes from the full log CPM data set. Because all of this, we did all our operations. We have now found out what are the genes that are on chromosome Y that are highly variant. Now I just want to see what they look like across these different samples. We'll see what this tells us. I know I've not told you the why so far, but that's part of the mystery. That's part of the intrigue. Hopefully you can tell me why I'm doing all of this and what the final plot here shows us. Okay, so far you're with me. At least it works without throwing errors. Are we good?
I don't see any questions in the slide, Google Doc, or in the chat. So yeah.
Okay. Excellent. So now what are we going to do? We have these 238, which are those genes across samples. I want to plot them. We know how to make the box plot, flip it, clean it up, remove the legend. We know how to do all of this, but we're just going to do it for the subsetted data here. So let's see what that looks like. There you go. Yeah, sure. We can change the name to something that makes more sense. But just as a quick reminder, what do we have here? We have the log CPM for all the samples for just the genes from chromosome Y that are highly variant. So this plot is showing us a much cleaner distribution than what we saw in the first box plot that we made with all of the data. So you have your samples and you have this. Now, based on the chromosome Y genes, what do you think this tells us? What do you think this tells us about the lower half of the sample or lower third of the samples versus these other ones? Anyone? Chat, please. What does this analysis, subsetting, plotting, and everything tell us about the samples? That's my question. Guesses are fine. Close guesses are fine. Okay. So, I didn't tell you where they got these samples from. Did I? I didn't tell you what was, were these from male or female samples? You don't know that. Or rather, I didn't tell you that at least. We probably don't even know that from the original data set. But we can infer it from the data. Because we have the chromosome Y gene expression values. Turns out when we go back and check, these are all the female samples. Right? These are all the female samples, and you have almost no chromosome Y highly variant gene expression that is going on here. And these are all the male samples. Turns out when we go back and check, this is somewhere in the middle. It's because they had pooled samples from male and female. Isn't this beautiful? You just got to this based on the data alone, based on your little data sleuthing that you did. And you didn't even have to know. You can just infer this from the data. You don't have to know what the biological sex was, and you can infer it from the data based on the expression. So this is how cool your initial, you just did what, couple of hours of data wrangling and visualization, and you've already learned something much more about the data than you could have ever fathomed when I threw that huge data frame at you, right? Okay, now I can't see all your faces lit up and happy, but I'm hoping that that's how it looks. So that's your really short intro to data visualization. I want to show you a couple of resources here. Let's see. So all of these resources are also available here in my data frame. Sorry, data frame. What am I calling it? My R Markdown HTML rendering, right? So these are a few. Wait, okay, cool. It's not here. I'm going to add it though. So one of them is called Data to Viz. I would totally encourage you to use it, especially if you're new to this whole business. First, forget about R or Python or your programming language. It just tells you, based on the kind of data that you have, what are the most sensible plots that you can make to interpret your data? Which is a pretty good start, don't you think? Even if you don't know programming, you need to know what plots to make wherever you're making them. So this is really helpful in that sense. Now, let's say I pick box plot, right? Then it takes me to something called R graph gallery. So R graph gallery and the corresponding, there is also one for Python. It'll explain to you what is that particular kind of plot. You can go back to datatoviz.com. These are a few examples. You'll see these are all ggplot. It comes with the gray background, right? But let's say I'm selecting this. The best part of it is it gives you example code, right? So you have something to start with. You don't have to figure out every single thing. That's not hard either, because people have had your questions, your errors, at least thousands of times before. So that's the beautiful thing about the R community. There's tons of answers, tons of support there on Twitter, GitHub, Stack Overflow, and everywhere. So you can do your Google searches or your Stack Overflow searches. Just say R Tidyverse your question, or R ggplot and your question, and most likely you'll have an answer. That is extremely helpful. Okay, so here you have example code. Now you know how to make sense of all of this, don't you? You know ggplot. What is the variable that it's working on? What are the aesthetics? Right? The theme. And then you're making a box plot. It shows you how exactly to do it. And for different kinds of box plots, and many times with different packages, not even just with ggplot. So this is a highly recommended source that I will use, and I'll also add it to the other helpful links part of it. The animations that you liked, the GG Animate, the Tidy Explain, all of these are also here. So let me go to the final part of our presentation, which is wrapping up. How do you save your data? That's it. I didn't show you much, but here we go. So you've saved see, here you have a few plot variables, and earlier on you created these other gene log CPM, gene underscore log. You also created these other data frames. Now you have to save them all. How do you do that? Again, this is part of Tidyverse. If you have a plot, right, you can just say gg save. Which is the plot? What is the name of your plot? Sorry, what's the name of the file? Here I'm saying I want to save it as a PNG, but I can specify any other format here. And if you want a publication ready one, which is 600 dpi, you can specify that. Specify your path, and you can save your file, and it will sit nicely here. Same way, you can also save your data. We already saw write underscore TSV, right, earlier on. But you can pretty much use, again, write underscore delim if you want to save a CSV, TSV, or something else. Again, what is the data frame that you're saving? Where are you saving it? Whether you want to save it with the column names. That's pretty much it. You can save all the data frame objects that you've created during your session as well, just so you don't have to come back and do all of it over again. Okay, and if you go to this HTML, which is basically generated from the R Markdown. If you just go to the R Markdown and say knit, you'll get the same HTML that I have here. So these are all a few helpful resources. And the best part of it is, look at all the stuff that you've learned today in this Tidyverse workshop. A whole bunch of functions, how to tidy up your data, how to wrangle it, how to visualize it, and how to save everything that you've done. These are a few resources that I've found to be extremely helpful. All of these are available for free as ebooks too.
Janani, sorry, you're not showing the slides.
Oh, I'm not showing the slides?
Sorry.
Wait, I can see the slides.
Really? I don't see the slide, so it might be from my side. Do you guys see the slide? Can anyone type in the chat if you see the slides, or do you see the console? Oh, okay. So it's just on my side. All right.
All right. Thank you. Okay. So these are a few resources. The R Markdown rendering was done with the distill package. I would like to really thank Arjun, who's another faculty here at MSU and also, well, my spouse. We generated the material together. And I would also like to thank Our Lady's East Lansing, Bangalore, Pune, ISCB Academy, all of you who have hosted me, because the material keeps getting better and fine-tuned based on how these workshops go. And if you need more such information, I would very strongly encourage you to go to the Our Lady's East Lansing GitHub page, because we have all of our workshop material freely available there for you to use. It is in data2biz.com, and Ralph, I will post it and update the GitHub right after the workshop. Okay. If you go back to the GitHub, it should have the updated information. The HTML, everything should have it. I'm sorry, it doesn't exist there already. And if you want to reach me, well, here I am. And by the way, along with this, I have a related piece of news. If any of you would like to reach out to me after August 1st, we'll be moving to the University of Colorado, Anschutz Medical Campus, to a brand new cool Center for Health Artificial Intelligence and the Department of Biomedical Informatics. So if you love R, if you love working with computational biology and you're interested in infectious disease, you know how to look me up. And with that, I'm done and happy to take any more questions that you have. Thank you so much.